Hi, so my name is Evan Gorstein, and I am a PhD student in the Solis-Lemus-lab at UW-Madison. And in this video, I'm going to talk to you about a recent publication from the lab titled HighDimMixedModels.jl, Robust High-Dimensional Mixed Effects Models Across Omics Data. So the context for this paper is the phenomenon of omics data or high-dimensional omics data in biology, um, where because of advances in biological technology, we're able to measure in parallel many different features of a biological system at once. And that produces what's known in statistical jargon as high-dimensional data. And so on this slide, you can see three examples of that. We have microbiome data, where you measure the taxa in an environment. A lot of different taxa can be present in an environment. In transcriptome data, you've measured the gene expression from a cell of a bunch of different mRNA transcripts. And in genome data, you've measured the uh, the nucleotides at different positions in the genome. Um, and high throughput data, in addition to being high dimensional, can also have batch effects. So we can have clusters of different samples, and we want to take into account those the clustering when we are fitting statistical models. And so if you're trying to do some sort of regression model, the standard way, or one standard way for taking into account batch effects, is to fit what's called a mixed effects model. Um, and this is sort of a schematic for what a mixed effects model looks like, where you have fixed effects beta and random effects BI. And the new challenge that we wanted to study when it comes to high throughput omics data is what happens when your fixed effects are high dimensional, and how can we fit these linear mixed effects models for high dimensional data. And so there have been a number of papers that have looked at mixed effects models for high dimensional data. And in our paper, we're drawing on the work of Scheldorfer and Gosch and Thoresen, who proposed this algorithm for maximizing the penalized likelihood of the model using the coordinate descent algorithm. And this algorithm, the basic gist of it is that it cycles through first the fixed effect parameters and then the random effect parameters. And for each component of both, it updates it to maximize the penalized likelihood. And so we've implemented that in a Julia package called HighDimMixedModels.jl. And this is a screenshot of from the documentation of the package. And hopefully it's pretty intuitive for how the interface works for our package. You just supply the function with the design matrices of the fixed effects, the design matrix of the random effects, the response, and the cluster ID. And then you also need to choose a lambda parameter, which controls how much you're penalizing the fixed effects. And you'd want to play around with that so that you get a sparse model, but so that you don't get all of your coefficients set to 0. Um, and we tested this approach on three different types of omics data sets, the ones that we mentioned at the outset, microbiome data, transcriptome data, and genome data. And we simulated our data sets to hopefully resemble data from these studies. And then we studied how the algorithm performs in selecting variables and estimating the effects of those variables. So for gene expression data, you can sort of see that the SCAD penalty does way better than the lasso penalty at selecting variables and also estimating those variables. And it's well known that the lasso penalty produces sort of a downward bias on the estimates of non-zero coefficients. And so we found that that was true as well in our mixed effects in the context of mixed effects modeling. Um, and similarly for the GWAS data sets, we found that the algorithm does a decent job of selecting the true coefficients while not selecting the non-true, the coefficients that are actually 0. And we see that with both the false positive rate and the true positive rate. And we found that just as with the gene expression data, the SCAD penalty outperforms the lasso penalty across a variety of settings, such as random effects covariance structure. And then we also simulated OTU microbiome data as well, and we found that, and we actually only used the SCAD penalty for this data, and we found that it does a pretty good job as well. One thing that I wanted to highlight as well was for the microbiome data, we included a variable that was measured at the level of the clusters, which is a nice thing that you can do with mixed effects models, is you can estimate the effects of variables measured at the group level. And so we found that we could also do that using our coordinate descent algorithm. And so just to summarize, we found that across all of these different omics data sets, the SCAD penalty was outperformed the lasso penalty at selecting variables and estimating those variables' coefficients. And we found that the number of false positives increased as a function of the degree of correlation between features, and also with respect, it also increased as a function of how complex the random effects covariance was. Um, and we found that unfortunately, if you use a lasso penalty, the algorithm is highly unstable, and you could end up converging to a sparse solution without setting every penalized coefficient to 0. Or the algorithm could not converge to a sparse solution without setting every penalized coefficient to 0 in some cases. Um, and then a few limitations that we also wanted to mention was that, well, I just mentioned that with the lasso penalty, the algorithm is quite unstable and can vary quite a bit as you vary lambda. And so you need to choose the right lambda, obviously. And that's true about the SCAD. That's true with the SCAD penalty as well. And we don't have any sort of quantification of the uncertainty in the parameter estimates or in the selection of which variables are important. And then the last one is that we don't have necessarily a way to choose which effects to assign random effects or which covariates to assign random effects, although typically you sort of know which covariates are going to have sort of different effects in the different batches from your data set. And so all of this and more can be found in our publication in PLOS Computational Biology. And this was co-work with Rosa Aghdam and Claudia Solis-Lemus. And we also have a link to our Julia package available right here as well. And so please take a look at your leisure, and I appreciate your time. Thank you.